So I'm pleased to announce our next speaker, Dr. Amy Vandergast. She is a research geneticist with the U.S. Geological Survey, and she'll be talking about harnessing local genetics. And when we thought about, that's a complex topic. Who could possibly talk about this topic in an accessible way? And we knew Amy would do it justice. So welcome, Amy.
Well, thanks. I am just thrilled to be here and be a part of the symposium and help honor Peggy and all of the amazing work she's done for plants. I have a confession to make. I am not a botanist. So thank you for welcoming me in to this. I do know about genes. And so what I'm going to talk about today is ways that we're using genetics to really help get the right seed in the right place at the right time. And I know many of you are not geneticists, and that's okay. You already know a lot about genetics. Humans have been selectively breeding plants as crops for almost 10,000 years. So we understand this process. And probably most of us remember learning about Mendel's peas and, high school biology class. And, so plants have been really critical to our understanding of genetics, throughout time. And Gregor Mendel is kind of given the name of being the father of modern genetics because he did the math to look at how traits are passed down from parents to offspring, and his study organism was peas. And maybe not everybody was super thrilled that he was focused on peas, either at the time or maybe even in high school biology class now. But based on those beginnings, we now have this information. We know that genes and genetic diversity is the foundation of biodiversity. So we heard earlier that plants were the foundation of biodiversity, but I respectfully disagree and think that it's genes and genetic information and genetic variation that really is the foundational pillar of biodiversity. It's the basis for selection, it's the basis for evolution of all of the diversity of species that we see and the ecosystems that they form. And those ecosystems are so important to us for the services they provide. So, along with our knowledge of how traits are passed down, we have had just a real evolution in our ability to survey genes in all living things. So, we are in the era of genomics. So, by the 2000s, the 2010s, sequencing capacity has really developed at a rapid pace. We can now survey whole genomes of organisms at the same cost and speed that we used to be able to only sequence a small portion of it. So, our ability to understand really what's going on at the whole genome level has really rapidly increased. And so, what that allows us to do is to survey genome wide genetic diversity of many individuals within a particular species. And that allows us to measure how related things are across the landscape and what the patterns are in that genetic diversity and how it's structured by things like gene flow, which in plants is the movement of pollen and seeds, and local adaptation to local environments. So I've had the privilege to be a small part of the Mojave Desert Native Plant Program, which is applying the National Seed Strategy for restoration in the Mojave. And as you heard earlier in Peggy's talks, there are these four major goals of the National Seed Strategy, and genetics fits into goal 2. So really, we're trying to provide information to help select that genetically appropriate seed as well as developing these tools in ways that help managers. And this has been a theme throughout today's presentations as well. It really just takes a lot of people from a lot of different agencies and groups to work together. And so, this is the list of partners that are involved with the Mojave Desert Native Plant Program under the direction of Judy Perkins at the Bureau of Land Management. And she's brought all of us together. And if I had put up slides here that listed names instead of organizations, I would fill the rest of my 15 minutes with all the people. So this is a robust, program that impacts a lot of people, that have a lot of input from a lot of people, and that makes it, I think, resilient. So in the Mojave, one of the main goals of native plant restoration is to help restore the habitat for this little guy, the Mojave desert tortoise, which is an herbivore and really needs native plants to survive and thrive in the Mojave Desert. And so the species lists of the plants that we want to grow and restore are basically built around this species as well as pollinators. So the real question is, why should we pay attention to seed sources? So we've talked at a big scale about why we should definitely take Mojave seed and use Mojave seed in the Mojave versus, say, using seed of the same plant species from Oregon or Washington. But what about within the Mojave? There's a wide variety of different ecosystems or ecotypes within the Mojave. There's extremes of temperature, high and low. There's mountains. There's valleys. There's a lot of diversity even within this ecosystem. So what we found is that basically 70% of desert plant species have shown some kind of home site advantage when they're grown in common gardens, and about 80% show adaptation to their specific climate. So basically, what we want to be able to do is use genetic data to help develop these seed zones that will allow us to plant seeds that are from similar locations and similar genetic backgrounds to the place we want to restore. And we want to be able to do this both for what we see and experience now, our current situation, as well as what's going to happen in the future with climate change. So this is an example of one of the plants that we've developed genomic resources for. And actually, this was a collaboration with the California Botanic Garden, and their geneticists did the work on this particular species. So this is the desert plantain. It's a wind-pollinated winter annual, and it's an important diet species for the Mojave Desert tortoise. It is found in sort of these meadows of plants in different areas. And basically, through our genetic analysis, was started by collecting at each of these locations throughout the Mojave, generating genetic data for multiple individuals from each of those locations, and then mapping that diversity along with information about climate. So, information about precipitation and temperature and other important variables. We can come up with a map. And so, this is our map that we came up with. And this is based on temperature zones that are linked to specific genotypes. So these are 8 seed transfer zones of increasing average summer temperatures. So the hot temperatures really drive patterns of diversity in this species. So here is the range of that, and I want you to kind of take a look right now at where we see the red zones. Those are really warm adapted plants, and where we see the blue zones, and then look at this next map, which is showing us where those zones are going to be under a high emission scenario of climate change. So we see a lot of expansion in the red zone, and we see a lot of loss in the blue zone. So this should be helping us to determine not only that we want to plant seeds in the zones as they occur now, but we also probably want to have some seeds from those warmer adapted zones that we're kind of throwing in the mix now to make sure that those areas contain the adaptive variation that they need to survive under warmer temperatures. So just to bring it back a little bit here to rare plants and also a little more locally to this region, I also want to talk just a little bit about how genetics can help identify unique diversity. And so one of the projects we worked on over the past couple years was to do genetic and abundance surveys of the Antioch Dunes evening primrose. And this is a very localized endemic. It's found only on the Antioch Dunes, which is up at the confluence of the Sacramento River. It's preserved. It's been listed as federally endangered since 1985. It's preserved on the Antioch Dunes National Wildlife Refuge, which I think is one of the only national wildlife refuges that is devoted to a plant. It's one of two plants that it was built for. And we went out and we surveyed the plants. And while we're out there surveying the plants, we're talking to the other botanists, and they're like, well, we've got this other primrose that's, kind of adjacent to this area. It's in some other patches. It looks a little different than this one. And we're not sure if it's this. Maybe it's this more widespread primrose that comes up through the Central Valley. We're just not sure. So we surveyed that one as well. And what we found through the combination of our genetic work and the morphology of these populations is that there are actually two unique subspecies in this area. And so we've now named another very locally endemically rare subspecies, the Bay Miwok evening primrose. And that was based on both genetic data and morphological data together. So I'm sorry, there's 72. It's not 71 that you still have to get. But yeah, so this is a way in which genetics can help us to learn more about our diversity and name our diversity so that it can be protected. And so that's what I have today. I provided just some of these QR codes. So these could provide some more information for you on both the Mojave Native Plant Program and then some of the research products that were talked about in this talk. And that's it. Thanks for listening. Oh, okay, great. I'm glad I didn't go way over, so I have time for questions. Yes.
When you're looking at genetics and you're trying to distinguish like a subspecies from a regional variation, I mean, I'm not a geneticist, but are there markers? Like, what's the delineation? Like, this is regional variation.
Okay, so the question is, if you're looking at defining a subspecies versus regional variation within the same entity. And I laugh a little because there's really no line in the sand demarcation for these things. So, I think what we try to do is make sure that you can see the difference. So you can see the difference in the physical makeup of the plants and some trait like the flowers or the leaves or the growth form. And you can also measure the difference in genetics. And the differences for the Miwok primrose were pretty distinctive, in my opinion, genetically. So it, it's a little bit of a gray zone, and that's because these things are not static. Evolution is a process. It's happening, right? As so there are things that are on the sort of beginning end of that journey, and some that are further along. Yes.
Regarding the Plantago, has the work that you've done informed seed collecting strategies from all the different agencies and groups, particularly emphasizing, the existing hot climate adapted locations and trying to really focus on them for future recovery efforts?
Yeah, do you think so? The question is, has the genetics that we've done on this and other species helped to inform the seed collections? And in theory, yes, it is. I don't know sort of at what phase this is happening, but Naomi might know. I mean, we use these maps and things.
Translate this to my phone, but so Judy Perkins will have us collect from those warmer seed zones as we're collecting seeds for particular species. So there is an emphasis on the warmer seed zones.
Good. Okay. Yeah. So an emphasis. So Naomi says there's an emphasis on the warmer seed zones. So yes, that's part of it. I'm sure you guys love getting out there into Death Valley and collecting those warmer seeds. Thank you for your service. Yes.
Hi, I work in commercial agriculture and we use like marker assisted selection for breeding. And one of the biggest challenges we have is that the library of traits is so small to be able to compare the markers from whatever the cultivar is to see whether or not it may have that trait and move it through the sort of breeding process. And I have been interested to hear, and maybe you have some insights on if we're going to be doing a genetic analysis of plants. Is there any thought in making that into like a database that can be accessed by universities so that we can start seeing how all of these traits, occur within the genome in a variety of species?
Oh, you've touched on a topic that's dear to my heart. Databases. Yeah. So the question is, are there trait databases that can be used with genomic data and that link traits in the genetic regions? Am I getting the question right? In development.
Building all this
Yeah.
using information, how do we share it and make a pool of resources to speed up and make that work
Yeah.
more efficient and easier and effective across different agencies?
Yeah, so I will say that all of our data go into public repositories. So all of the genomic data that's collected for every individual that we touch goes into GenBank. We have USGS data releases that release derivatives of that data that might be, more appropriate for someone to use. Collaborators are creating map tools that restoration groups and growers can use to see what zones they should be transferring between. And I think that our knowledge of some of these native species is well behind, what's happening in crops. So for a lot of these, we don't have a full assembled genome, to compare things to, and we're kind of looking at this through a lens of finding gene regions that we can tell are under selection, and then looking at correlations with the environmental variables with those genes. And so we're at that stage. We haven't really mapped any of these regions to know, what is the pathway that they're involved in, and how are these traits evolving. So it's still a very coarse scale. I don't know if there is a database for traits for plants, but I'm going to look into that. And
There's not on the
Okay.
website yet.
Okay.
So
Yeah.
probably-
Yeah, but
we did.
yeah, I think it's always been a tenet of our science within USGS that the products that we produce are public. They are paid for by all of us taxpayers, and so there's a big emphasis on making sure that those data are available. Yes.
So climate change is happening quicker than what nature could do per generation to adapt to a higher UV and warmer temperatures. In your research of collecting plants that have adapted to a warmer and or drier area, are you trying to isolate those characteristics, isolate those genes that perhaps there's a sub-study that could do splicer or something that allows a bigger variety?
Yeah. So the question is, since we are warming our planet at a level that's unprecedented, much faster than anything that's happened in the past over evolutionary history, then why, or are we trying to isolate these traits and use techniques like CRISPR or something to like move genes around? I am not involved with anything like that. That is, there might be some of that work going on. I don't think it's necessarily happening right here in our Mojave plant program, but it is something that I think researchers are talking about and thinking about in other systems. I think we do have some kind of things like that going on in USGS with relation to specific invasive species to try to help deregulate how quickly they can reproduce and survive in areas, but it's all very experimental right now. Okay.